Okay, shall we start? All right, good morning. Second to last day. Everybody still awake and with us? Okay, how was the poster session yesterday? Yeah? Some of you are a little bit shy. Please do present. We want to know, and everybody else would like to know what it is that you're doing. So don't go hide in some corner, come talk. Okay? All right, good. So today is going to be a research talk where we'll draw upon and use some of the concepts that we have learned so far already. And I'll tell you some work that we have been doing about which I am super excited. This is really, really interesting and in my mind something very different that I did not expect to find at all. So what is this? I'm going to tell you about the evolutionary impacts of mutation rate and spectra. And the main question that we're motivated to ask in general is how do organisms adapt to their environment, right? This is something that in general, as evolutionary biologists, we want to understand. Here are some examples from a recent review paper. I just like the drawings here of cases which have been very well studied where the genetic basis and the phenotypic basis of adaptation to new environments has been very thoroughly worked out and continues to be worked out. So these are cases where these two mice are very closely related groups of mice and they live in different environments. One lives in a desert habitat where the sand is white and one lives in a normal soil habitat where the soil is dark. And so they have adapted in using many different loci where their coat color has changed over time to be better fitted or matching with the environment in their background. So it's for camouflage. This is the classic case of the peppered moth, who most of you hopefully have heard about or read about at some point, where industrial pollution selected for this melanistic moth version over time. And now after some areas were cleaned up in Manchester, which is where these big mills were first, where the soot was now removed and reduced over time, there was again some selection favoring the other one again. So this is very interesting, and the genetic basis of this has also been worked out in the last few years. Sticklebacks are also a very nice system where people have studied what has allowed these marine stickleback populations to invade and colonize freshwater habitats multiple times. What are the alleles that underlie these adaptive events? And again, they map to a few different very big effect loci, which people have studied a lot, involving the armor plates and the body shape and size. Because in the marine environment, they don't have the same kind of predators. They have different constraints based on salt water, mineralization, and so on and so forth. And then finally, this is an example of mimulus, monkey flowers, where pollinators have driven divergence between these different flower phenotypes because different pollinators in different areas are doing the pollination for these flowers. And so to cater to hummingbirds versus cater to bees and other insects, differences have evolved. And again, the genetic basis of this has been worked out. So in general, now we'll get a little bit more technical. I don't think anybody has shown you adaptive landscapes yet, right, in this fitness landscapes. So this is one way in which we can think of, as at least in cartoon form, how organisms or populations traverse fitness peaks and valleys, okay? So when we're saying a population is adapting, if we plot genotype space in some abstract way along these two axes, and fitness is on the third axis here, we can imagine there are these peaks and valleys which represent areas of high fitness and areas of low fitness given a particular genotype combination, right? So you can imagine this is one piece of DNA, this is another piece of DNA, and the combined genotype sequence at both these loci determine fitness, for example. Everybody with me so far? Yeah? And so when a population is starting up, let's say from here where its fitness is low, and it's acquiring mutations randomly to begin with, right? And mutations that are adaptive will allow it to slowly traverse some peak and walk towards a trajectory where selection is driving the population fitness to increase. And you can think of it as populations climbing some peak, okay? This is not a perfect representation. There are issues, but we won't go into that right now. This is a useful starting cartoon to at least think about how populations evolve in genotype space. Now, what determines which kind of mutations are going to be observed during such adaptive processes, right? So first thing that should come to mind with what you've learned already is fixation bias, which depends on fitness effects of mutations as well as the the population size, right? So imagine there are 3 different populations that are evolving and adapting in the same environment. And in each case, we're finding that the purple allele is fixing or is at high frequency, okay? So the first thinking might be that the purple allele is very beneficial, right? And maybe it fixes again and again in these populations because these populations are large. They're able to find the best, the fittest allele and sort of fix it. Everybody with me so far? And so this kind of bias is called fixation bias, where highly beneficial alleles are more likely to fix in populations. Correct? Okay. However, another factor that's also important to decide which kinds of alleles might be fixing in populations is mutation bias. What do I mean by that? Certain kinds of mutations, if they're more likely to occur in a population, given the biochemistry, given the DNA, etc., then those mutations are also more likely to be observed in the population at the end. Of course, they have to be beneficial, right, if they are adaptive mutations. But of possible other beneficial mutations, you might have the purple one fixed not only because it is high benefit, but also because it occurs more often. Simply a sampling effect, okay? So this effect of mutation bias was kind of ignored for some time in between, and now it's making a resurgence, and there's a lot of data slowly as we have more information on exactly which mutations are available to a population, because in between we couldn't do those kinds of analyses given our inability to sequence and sample enough mutations in populations and determine what the mutation bias is. Now it's becoming very clear that mutation bias can have a big role to play in adaptation, okay, and walk you through some of the evidence from recent work. So what kinds of effects have we observed? It can determine genetic basis of adaptation. I'll show you a couple examples later. As a result, it can drive parallel evolution. So you've heard this now, if something happens again and again in different populations, the same mutation is fixing, the same phenotype is evolving, the first thought is like maybe it's because it's beneficial, right? That we take that as a signature of adaptive evolution if the same thing is happening in multiple populations. Everybody with me? So one important factor that has come up now though is that parallel evolution can be facilitated more if the same kinds of mutations are likely to happen in different populations as well, right? So again, I'll show you some examples of this, and this is also important in the context of predicting drug resistance mutations. So it's a very nice study that came out. This is the one I'm talking about a few years ago, where they looked at some tumors, different kinds of cancers in large patient populations, and they identified which mutations were responsible for drug resistance, right, where these tumors were not responding to drugs that were given. And so drug resistance had evolved, and they asked, if you want to predict how likely is it that a particular mutation will come up and be observed in multiple patients independently, turns out it correlates well not with the fitness benefit of those mutations compared to others, but how likely those mutations are to occur in the human DNA to begin with, right? And so let me describe a little bit more. Before I go there, a little bit more about work that we have done and things we've been interested in. Mutation bias can also help to explain many genome-level traits. This is work that encompasses a lot of different literature over many, many years. So genome GC content, which is how many bases in your DNA are going to be, what fraction is G or C? Yeah.
First of all, I think I know that you speak about mutation bias, but I just wanted to clarify this point. So, you just mean that these mutations are asymmetric. For example, if I just, that has been done for a long, long time, so what's new here?
I'll come to it.
My feeling is that, so I just tell you what I feeling is, I thought mutation rates and selection are anti-correlated. And that's what is mutation bias, or maybe this phrase that you are using and others use, is it different?
Mutation rate and selection coefficients are anti-correlated, yeah, but that's not what I'm talking about in terms of mutation bias.
Asymmetry in the mutation rates. 0 becomes 1 with some rate and 1 becomes 0 with some other rate. And that's sort of general, and it's been there for lots of models, so I'm just confused. How is it,
So there's been very little data supporting this though, right? That it doesn't matter first of all, right? And it's not just 0 to 1 and 1 to 0. It's also
everywhere.
All kinds of mutations, right? And so if some kinds of mutations are more likely to be sampled, to happen in the population, those are probably more likely to also lead to beneficial mutations which are going to occur. And so that is the, how much does that matter for what you see?
In the DFE, right? So the DFE that you see, if it has a high selection at some, let's say 1.1 is high, that means those mutations were more probable. So I'm saying the mutation bias, won't it show up in DFE?
No, right? I mean, not necessarily. It depends on how you're getting the DFE, right? If you're looking at the DFE as for mutations which have occurred already, then your DFE is already skewed to reflect that mutation bias.
So that's the fixed figure, sort of, the fixed DFE, the fixation one, and the ones before fixation.
Yes.
Right?
So,
before fixation, people have measured, no?
Right, but people have not asked if the mutation bias matters there. So let me give you an example, right? So let's say we take, I'll show you some data which will talk about this, but let's say we're dealing with A to C mutations and G to A mutations, okay? Just two kinds of mutations we're going to classify into. And we do the DFEs of both of these. So if the DFEs are not looking the same in both cases, then it might matter for which kinds of mutations are going to be reflected after selection. But even before that, if 90% of mutations are this and only 10% are this, then even if there are a bunch of possible beneficial mutations which might be, the population is never going to see them because these mutations just don't happen as often, right?
So this bias has already come in the DFE, right? The distributions look different. And therefore, what happens in adaptation, these DFEs are going to get in and will affect adaptation. So if I take these DFEs, I will run a simulation, calculation, whatever. Right? So there is this mutation bias coming, is the shape of the DFE.
No. How often are you going to even encounter this DFE at all? So if you're going to be sampling from some general DFE, let's say, right, when you're doing your adaptive walks, you have to sample from both these DFEs in the proportion in which these mutations will occur, right? So if these mutations are 90% frequency and these are 10% frequency, you're going to draw 90% of your mutations during adaptive walks from this DFE and 10% from this DFE, right? And so that is the mutation bias that I'm talking about. That for some reason, if certain sets of mutations are more likely to occur, they are generally more likely to be involved in adaptive processes simply because they don't occur, there are others.
Okay, so some mutations, they will not just be asymmetric, but also the DFEs if I'm using 0 to 1, I have some mutation rate and I use like a DFE, but 1 to 0, I should use different DFEs. Is that the point?
That's possibly the case, right? Or even if the DFEs are not necessarily entirely different, well, actually, no, unless the DFEs are different, it won't matter. Yes, you're right.
So that's the sort of the pattern.
Yeah. And we don't know first whether the DFEs are different. So that's measurements that people have been trying to make now. And second, even in theory, how much this difference can matter, forgetting even if the DFEs are similar, just in terms of which kinds of mutations you'll see later, will change based on this, right? And so that theory also people have been developing over time, but has simply not gotten as much attention, I think, until now. And so in the last decade or so is where we have a lot of data suggesting and enough evidence suggesting that these kinds of asymmetries in which mutations will happen at what rate, it matters a lot more. Okay, that's only come up recently. Okay, so maybe I'll skip this part actually because it's not that important or relevant. So here is the kind of mutation bias I'm going to talk about today. So you know there are 4 bases and there are transitions or transversions. Roughly we can classify mutations in 2 categories. Transitions are things that take you from purines to purines and pyrimidines to pyrimidines. Everything else is transversions, okay? And so as you can see here, if you just have a simple unbiased set of mutations, if all types of mutations are equally probable, then you expect to see that in an unbiased state, 2/3 of all mutations should be transversions. Everybody with me so far, right? So if nothing else is going on and if mutations are entirely random, you expect to see more transversions compared to transitions, right? But what we actually see if we look at organisms and what these mutation biases are and how we have measured this, I can talk about it later. But just say that somebody has gone in and measured 4 different bacterial species, how many mutations that occur before selection acts on them, okay, just baseline, how many are transitions and how many are transversions. And universally we find every taxon that people have looked at so far, not just bacteria, there are many more transitions than transversions. Okay? There are only a few species where that ratio is 1:1, so it's 50%, that's here. Okay? And so there's nobody who's on the transversion side, even though remember we expect there should be more transversions if everything else is equal. So this has been a puzzle that people have thought about for a while and there's no answer. Nobody exactly knows why this is the case. There are many suggestions people have had. For example, if you look at the codon table and calculate, if I get different kinds of all possible transversions, all possible transitions, how many of them will change amino acids dramatically, right? Go from one corner of the codon table to another and so on, which you would expect, as we discussed yesterday, would lead to very different amino acids, right? Like big, bulky ones replaced by small ones, vice versa, biochemistry changes and so on and so forth. So people have suggested that transversions should be perhaps under selection. What kind of selection? Negative selection, right? And they will be thrown out of the population because they are more likely to lead to different kinds of amino acids, okay? Big dramatic changes in amino acids. And so when people have asked in evolution experiments, what do we find, or evolution studies, are there more beneficial transitions than transversions? There isn't, okay? There's no evidence for that. So then that hypothesis doesn't fully make sense. In any case, it doesn't explain why even baseline we don't observe many transversions. Why are there more transitions? This is probably something to do with the DNA structure, the biochemistry of how repair and replication occurs, how mutations occur, but we don't have an answer for this yet, okay? But so this is a fact at the moment that most species we know of have transition bias. But what's also important to notice is that the nature or degree of that bias is also quite variable, right? There are some species that are much more transition biased, some species are much less biased, closer to an unbiased state. Why do we see this difference? Right? What causes this variation? The fact that there is such variation across species suggests that across phylogeny, right, across lineages over time, some species move from here to here or here to here and so on. Why do they move? How does that happen? Is it happen under selection or is it drift? And so we wanted to address these kinds of questions. So we wanted to ask, how do mutation biases impact evolutionary dynamics? What do? We collected random samples of mutations in bacteria with different mutation bias. So here's the wild type E. coli, which has a transition bias, as I just told you before. And we allow single lineages of these to evolve in mutation accumulation lines, right? So we propagate single cell bottlenecks, as I explained already in previous talks. And at some point, some mutations may happen, shown here in gray, at some rate. Like we can predict based on the known mutation rate for E. coli, okay? We can then also do the same thing for a strain of E. coli where we've deleted a DNA repair enzyme in that background strain, the wild type strain. This is well characterized already. So the MUT-Y gene is a gene that encodes for one particular DNA repair enzyme in the pathway. When we delete it, you increase the mutation rate, but you also increase the rate of transversions. Okay, that's it. That'll become important later. So what's going to happen? There's going to be a higher rate of mutations accumulating here. And we can pause these lineages and sample them at specific time points where we predict that a mutation on average should have occurred in a lineage, right? We will of course be wrong a lot of the times because this is just a prediction. And mutations are going to happen randomly without telling us when they happened and without knowing that we're predicting something. So we sequence lineages at different time points in these populations. And we identify strains with a single mutation. And why single mutations will become clear in a bit, okay? So there'll be a bunch of lineages we sequence which had 2 mutations, 3 mutations, 10 mutations, whatever, compared to the ancestor, okay? We don't use them for this analysis. We focus only on the ones that have a single one-step base change compared to the ancestor. And then we measure the effect of each mutation using relative growth rates, right? So we're measuring the growth rate of these ancestors and asking each mutant here, how much has it changed the growth rate of this ancestor? Okay, so that gives us an estimate of the fitness effect of that mutation. and then we can build a DFE using it. Everybody with me? Okay, good. Now we have to also correct the distribution of fitness effects for some selection bias that can arise during colony growth. I'm actually going to skip this right now as well because we're going to run late otherwise. If we have time tomorrow, I'll tell you a little bit more about it. Okay, so what do we find, right? Forget the correction, whatever, doesn't matter too much right now. So the MUT-Y DFE we find skews beneficial in many environments. So here's the DFE, you've already seen DFEs of various sorts. Again, the way we're calculating fitness effect here, 0 means that there is no effect, okay? So these are neutral mutations. This is the frequency on the Y. In pink is going to be the mutator, the MUT-Y genotype, and green is wild type. In two environments, we find that the MUT-Y strain has fewer beneficial mutations compared to the wild type strain, okay? Remember our null hypothesis is that if these DFEs are not different, they should be, most of our environments, we should find their overlapping DFEs. And we find that only in 5 out of the 6 environments, or 16 environments in which we tested these DFEs. In most environments, what we actually find is that the mutator DFE is shifted towards beneficial side, okay? And so the mutator here, in this case, so imagine this is the mutator, this is the wild type. So this is sampling more transversions, this is sampling fewer transversions compared to transitions, and we see this kind of effect in many of the environments. This was puzzling to us, okay? What is going on? Why should this be the case? Yeah.
So again, it reminds me some of the older work, I think, Madhava and so on, where I think they measured DFEs across various environments, like good environment, pretty bad, pretty good, and DFEs were different.
Yeah.
Right? So how is this different? What does it
No, this is where the DFEs of these two strains are different, which you wouldn't expect. You would, of course, expect some effect of environment on the DFE, right?
Yes.
So for the wild type alone, if I compare DFEs across environments, they change a little bit here and there. That's expected, that's known, that's fine. It's the difference between the wild type and the mutator DFE, which is also environment dependent. And in most environments, the mutator has a DFE which is right shifted compared to the wild type. And that overall consistent effect is what is puzzling, right? Because I would have expected either that they are the same in most environments. There's no reason to expect these DFEs should not be the same, right? Going in. And if they are different, why is it that they're different in one direction? And that's the puzzling part here. Is that?
Okay,
Okay, yeah. So,
if I had activators, you expect everything to be with the opposite side?
not necessarily. So I'll get to that, right? So this is the basic observation, the pattern that we were kind of struggling to understand because didn't expect that necessarily, right? And we couldn't understand why. So we tried to ask, okay, what is different about the kinds of mutations that have occurred, right, in the genomes of these strains that we've sampled here? Is it that the mutations are in some weird genes in some cases? Are they on the leading strand, lagging strand, synonymous, non-synonymous, coding, non-coding? None of that correlates with these differences that we see in the DFEs, okay? There are some differences, of course, in the wild type and the MUT-Y. And remember, we have only 80 mutations that we're sampling across the whole genome, wherever they occurred, right? So our power to detect any big impacts of exactly which gene, exactly what, is very, very low. We don't have statistical power for that. We simply don't have enough mutations.
So, like, so I was surprised to see lethal mutations for the mutator, right? -1 is a negative.
No, that's actually the wild type, not the mutator.
Orange ones.
Orange is,
The first 2 environments.
yeah, I mean, they're not lethal, but they're deleterious, yes.
Yeah, so why is the wild type lethal and then mutated does not and goes away?
That's just, no, so it's not going away. So these are different sets of mutations that are sampled by the wild type and the MUT-Y, right? We just did an MA line and they happen to sample whatever they happen to sample. So what's happened is that also, so you see lethal mutations here because we evolved these, did these MA lines in LB, okay, in a different media. And so there, of course, if somebody was dead, then they was dead. We would not sample it, right? But those guys there were clearly some mutations whose effect in LB was not lethal, which is why we could pick them up. But you put them in some other environments and they're lethal. These guys don't grow at all, okay? So that's fine. Okay, so let's go on. And this is where we started talking to Lindy, a mathematician, who was at the popgen school last time. And we ended up collaborating with her, and she helped us figure out what is going on here, okay? So what is going on here? Overall, we see more beneficial mutations in MUT1 if we combine data across all these different populations. Remember, what's happening is that even though we're calling this the mutator, we're still sampling only strains which have a single mutation, okay? So mutator, should have a higher mutation rate, but that's not playing a role here at all, because we're still sampling only every strain that has a single mutation. So why are there fewer beneficial mutations in wild type? And before I get to that explanation or whatever we figured out, I want to also highlight a few other things that happen as a result of this DFE shift. So we have not really talked about plyotropy very much in this course so far. But if you imagine two environments, environment 2, environment 1, on which we are plotting relative fitness, okay, along these axes. In general, we are interested in knowing how often will a mutation increase fitness in one environment but decrease fitness in the other environment, right? This is called antagonistic plyotropy. That would be these yellow cases here, where fitness in one environment is increasing due to a mutation, but fitness in the other environment is reducing, okay, or vice versa. And these cases are interesting for evolutionary biologists because these represent situations where you might see a trade-off, correct, due to a single mutation. Like one mutation that is beneficial in glucose may be deleterious in lactose, and that's a problem if the environment is going to fluctuate back and forth between glucose and lactose, right? So these kind of trade-offs have been interesting. So we want to understand, is there a difference in these kinds of pleiotropic effects that we observe in the set of mutations we have? The opposite side here, which is of interest for adaptation, is also beneficial synergistic pleiotropy, where mutations are beneficial in both environments, okay? Because then it means that, okay, if you adapt to one environment, those same mutations could also allow you to adapt to, could be already adaptive in another environment. These would be really fun to have because then it makes it easier for the population, even if the environment is changing. So we asked, how many such beneficial synergistic pleiotropic situations do we find? And again, the MUT1 strain has many more of these. So 12% of all mutations that we observe are beneficial in two environments, okay, pairs of environments. And that fraction is much lower for the wild type. So there are effects beyond just that one DFE that I showed you. What's also interesting is the number of environments in which we see consistent fitness effects. So let's say I pick out a beneficial mutation in the wild type in glucose. I ask, is that beneficial in all the other environments, okay? So, and that's what's plotted here. How many mutations do I find where a given beneficial mutation is beneficial in 10 environments, 15 environments, and so on? That's what's being plotted here. So for the wild type, if you pick beneficial mutations, you find that most of the time they're beneficial in only 1 or 2 environments, okay? You don't find cases where they're beneficial in many environments. In contrast, if you pick a deleterious mutation, let's say deleterious in glucose, I find that it's actually deleterious in 5 other environments, most often, okay? And of course, there's a spread in this distribution. For the mutator, on the other hand, the pattern is reversed. So we find that deleterious mutations are uniquely deleterious in a few environments, but beneficial mutations are beneficial in as many as 5 environments, right? So a chance that a beneficial mutation in the mutator is going to be beneficial in other environments is higher, okay? So the prediction based on all of this is the mutator should have a better time of adapting, okay? Even based only on these DFE and so on, without taking into account its mutation rate at all. And we'll come back to this later. So then what is going on exactly? What's the basis of all of this? So I told you before that the MUT1 samples many more transversions as well as GC280 mutations. So that's shown here, okay? And the median fitness effect of a transversion is about -1%. So in general, remember, mutations are still deleterious on average. And transitions are a little bit more deleterious, okay? So 3% fitness decline. And some similar differences observed for these class of mutations as well. So this is where Lindy did some adaptive walk simulations to ask if these patterns are general and when are shifts in mutation bias beneficial. So let me, yeah. Tenfold, yeah. Okay, so what happens here in these models, this is a little bit more advanced, and I don't think we're going to get into this Kavita, at all, right? These kinds of models or adaptive walks, are you going to? Okay, so I'll just briefly explain what the general logic is and point you to papers if you'd like to read more. What you're doing is you're laying down a fitness landscape, okay, at random. You're saying, okay, I'm going to take a string of, let's say, 10 bases, which is my sequence of DNA. I'm going to arbitrarily assign fitness values to possible genotypic combinations I could have, right, of when mutations occur. So let's say AGGGTTTT CCC is going to be here, okay. Some other fitness value is assigned to. Basically, each genotype gets some random fitness assigned to it. So as a result, your fitness landscape is very rugged. Lots of peaks, ups and downs, and so on. Okay, so Lindy sets this for every simulation, and then plonks the wild type down at some point, again randomly in the landscape. That's the genotype it starts with. And given that genotype, it starts with some fitness. Okay. Now, she samples a random set of one-step mutations that are available to it and asks, of all possible one-step mutations given the genotype space which it occupies, what is the DFE that would construct from that? And you can construct a DFE because you have that fake landscape you've put down, correct? So given that landscape, given this is where the genotype is, if I sample some 300 mutations around, which I can access in a single step, what is the DFE of those mutations, okay? And so she keeps track of these DFEs. Now from that DFE, she's going to draw one random mutation. If it is beneficial, it is allowed to fix. If it's not beneficial, it gets chucked out, okay? So in this process, there's a walk that happens. Population goes forward by one mutation at a time. Multiple mutations are not allowed in this initial set of simulations. And then a beneficial mutation is fixed, another beneficial mutation is fixed, and so on. And every few generations of these walks, she pauses and asks, what is that DFE looking like, okay? And how many beneficial mutations are we seeing in that DFE? So what's the fraction of beneficial mutations in that DFE? Yeah?
So at every step, we find the DFE available and the DFE fixed.
Yeah.
At every step of
Yeah.
the walk.
Yeah. Yeah, I mean, I don't know that she keeps track of it but the point is that, yeah, that happens, yes.
But the available one probably will not change. The fixed one, we know that it changes like the Garish-Lensky's work.
How?
But the available one,
So the available one will not change, meaning?
at every step of the walk.
It will, right, because the
So take the data, every step, take one step, I sample it, consider took the one of the pieces, and looks at its neighborhood
Again,
put
right.
all that data in the
Right.
same histogram. Okay, construct a new histogram.
Correct.
So I expect that they will be same. So are you saying they will not be?
Maybe they're more or less similar over time, but it depends on what the landscape is exactly and where you start and whatever is going on, right?
Yeah.
But let me come to what happens, yes. So you do this basically throughout, okay? And you allow, let's say, the ancestor, the wild type, to just evolve by itself for some time, okay? Now what we're interested in is what happens if you have a bias shift, okay? Now when there's a bias shift, what effectively is going to happen is instead of, let's say, the wild type is sampling this space because these are transversions on this side, okay, of the genotype space. If you have a bias shift, you now have the ability to sample mutations in this area as well of the landscape, right? Because now the MUT-Y is sampling many more transversions. Got it? And so effectively, you now constrain your sampling of mutations based on whatever bias we know exists in these populations. So when the wild type is allowed to evolve in this simulation, it evolves, but it's sampling transitions and transversions at some ratio which we know, correct? So it's sampling more transitions than transversions. So of the available single-step mutations, they're going to be sampled in the proportion that we know of, okay? But that DFE may or may not be different for those depending on what the landscape looks like, correct? Because the DFE here is not of a known fact, it's being derived given the landscape, right? So in the wild type, these proportions are more balanced a little bit, but in the mutator, this is a very stark difference like this, okay? The mutator samples 95% transversions, more or less, compared to wild type sampling 45% transversions.
Where is this transversion and all of this landscape?
In the genotype itself, right?
Okay, so this is, each side we have 4 bases, is that one?
Correct, yeah, Yeah. And so Lindy has also done versions of these simulations where she does amino acid specific simulations, various codon bias, codon models, and so on and so forth.
So what's the
But
model again? So for the wild type, she has some A to C and G to A rates.
yeah,
And when you go to mutators, you
those change, correct? Yeah.
That's all. That's all.
Yeah.
So the change is actually just in the mutation rate, no?
In the relative mutation rates, yes. And so also in the simulations, the overall mutation rate is not different, right? So we're simulating a bias shift, but we're not letting the mutation rate, total mutation rate change for this.
I can understand.
The overall mutation rate,
Overall means what? I mean, so wild type
ha,
is going A to T, A to C, C
Yeah.
to A, and so on.
Yeah.
So that's some mutation rate, which is the total one.
Yeah.
And for mutator, everything is 10 times. Is that
No, that's what I'm saying. That's not happening. The mutator is also evolving at the same rate. So
So where's the mutator?
it's not there. That's the point. I just call it the mutator because, yes, sorry, but it's just a MUT-Y strain. Like, forget it's a mutator at the moment. Because we want to understand what's the effect of the bias shift, right?
That's I think that's what I'm again missing. So for bias shift in the model, because I think I can understand model better.
Yes.
So then what happens in the model?
So,
You simulate the shift. I think that's understood.
right, so there are some rates at which mutations can occur, which is where the sampling is coming from. Every time a new mutation is being sampled, so that DFE that's being constructed, right? And from there, one mutation is going to be sampled and fixed ultimately. So the sampling of mutations is where the bias shift occurs. So in some cases, for the wild type, let's say, all of these rates are imagined similar. But for the mutator, some of these rates are very, very small, right? And so these kinds of mutations are less likely to occur. So what it's going to end up doing as a result is, let's say the wild type is they're starting off here, okay? And let's say with the wild type mutation bias, which is tilted towards one type of mutations, you'll end up sort of sampling this area more. In the mutator bias, you might end up sampling this trajectory more. And that is the key difference that is possible to happen, right? And then the question is, given some random fitness landscapes where we have not made the landscape one or the other way, is there an overarching difference that shows up in these kinds of trajectories? Okay, and what is that difference looking like if there is a difference? So what is plotted here on the y-axis is the difference in the fraction of beneficial mutations. So basically asking, every time we sample that DFE, what is that fraction of beneficial mutations in both of these strains? And that difference is being plotted here. And the difference is plotted so that any positive values here are going to indicate that the MUT-Y strain has a benefit, in the sense that it has more beneficial mutations compared to the wild type at that point of time. And negative values would mean the opposite. On the x-axis is the bias shift that we're simulating. On the left would be more transitions, right is more transversions. And to orient you, this is the no bias situation in pink, right, where 66% of mutations would be transversions. Here's the wild type bias that already exists, which is, like I said, biased towards more transitions, right? So the gray is really the starting point, the ancestor, where we start sampling mutations. Now what Lindy is doing in these simulations is, after some time of the wild type evolving in that landscape, then a mutation bias shift is introduced, okay? So she asks, if I introduce that bias shift at different points of evolutionary time of adaptive walks, does that matter as well? And I'll come to that later. So what you're doing then is, you're simulating a shift where you're suddenly now going towards the MUT-Y bias, which is very close to almost 100% transversions. And the top half is going to indicate, so any points that lie here indicate bias shift to be beneficial, correct? Because you have more access to more beneficial mutations. And this would be a case where the bias shift is deleterious. And what she's asking is, I could go from this wild type bias in all possible directions, right? So we simulate that. We simulate cases where you go all this way. We simulate cases where you go just a little bit this way, and so on. Okay. Any questions about this? It's a little bit complicated, I know, but okay. So what do we actually see? Okay, ignore the colors for now. Focus on one line. I'm going to take this light pink line here. Okay, what does it mean? It means that anytime you're going from a wild type bias, which is in gray here, right, to a bias which is a no bias situation, pink, you're going to end up sampling more beneficial mutations, right? Now, if you go further towards the MUT-Y bias, that sampling is going to be even stronger and even higher, correct? So now if you look at all these different colored lines, the colors indicate how long was the wild type evolving in that landscape before we forced it to move its mutation bias. Anytime you move across these different regimes, I'll explain what they are in a bit, you get a benefit for the MUT-Y strain, for the new biased strain. In contrast, anytime you move to the left of the gray line, you're going to get a reduction in available beneficial mutations. Okay? Making sense? So what are these different colors? They indicate later during adaptation, the light colors, and dark colors are early during adaptation, right? So in an adaptive walk, what we generally expect to see is over time, fitness will increase, right? This is similar to what we saw in the Lenski lines, remember? And so we're asking, if I now simulate a bias shift here, which is early during adaptation, what's the impact? If I simulate a bias shift here, late during adaptation, what's the impact? And what you predict from this simulation is that early during adaptation, if you do a bias shift, there'll be a small effect, okay, a small benefit to be had. But later during adaptation, there's a huge benefit to be had. Okay, so light pink lines are much bigger. Same way, if you have a bias shift which goes and makes the existing bias more extreme, right? So you're going to the left of the gray line, you're going to get a bigger problem and a drop in beneficial mutations later during adaptation. Okay, so light pink line is at the bottom, yeah.
This later is because this is fairly rugged landscape.
Yeah.
And you get stuck in these local peaks, and therefore it's good to have the bias and allow more better sampling.
No, it's more that you have fewer beneficial fraction available later during adaptation.
Yeah.
And so later during adaptation, because your existing bias allows for a small fraction of beneficial mutations, a bias shift is more beneficial because it allows the sampling of a different mutational space.
It should be more important in rugged landscapes, because you get stuck in local peaks. Rugged landscape, you just take this single peak landscape.
Yeah, I don't remember how the results changed exactly with more ruggedness in the landscape. So she did those simulations. I think the overall patterns were not different, but I don't recall now whether what you're saying happens or she was observed or not.
There's a counter argument also. So
I don't know which way it plays out, but she's done those simulations. I should remember the supplementary figures here, but I don't. Yeah. Okay. I know some people are confused. Can I help understand this better? So let me give you some intuition, which is in the next slides, right? What's going on? So imagine with these DFEs, so now how do we connect this to what we're actually observing in the experimental data, right? What we think is happening is, if you think of the pie as showing you a different way of seeing how many, what the DFE is, right? So gray is deleterious or neutral mutations, and there's a big chunk of beneficial mutations in this DFE, okay? Now, these are all already sampled if your population has been evolving for a long time, right? Because as it evolves, it's going to keep sampling beneficial mutations, keep fixing them, right? The red slice indicates what is still available because it has not been sampled yet. Now, what we're saying is happening is if your bias shifts towards a space in mutational, part of the mutational space which has not been sampled before, such as transversions, with a very strong bias towards transversions, then it's not that the overall pie changes. Overall, there are still just as many beneficial mutations, but what you have already sampled is a very small piece of that pie, right? And the available mutations in red is much larger, simply because you have not sampled transversions before. Okay, to make it even more helpful, so this is one of my favorite movies, and it's about these gold diggers, right? People who go and used to go and pan for gold in rivers and so on. So those days, this old man in this movie is like panning for gold. You basically go in these river beds where there's supposed to be more gold, and you look and you look and you look, and maybe you find some gold nuggets. The bigger, the better, of course. So if you keep looking in the same places where others have also looked, there's a big crowd of people looking for gold in the same places, you're not going to find very much because people have already found the big stuff. But if you go somewhere else, far away, where nobody's gone before, you might be very happy because there you might find these big chunks of gold just because nobody has looked there before, right? And so I think this general intuitive idea is we now have a mathematical framework to understand it, think about it, and so on and so forth. It turns out it's very, very simple actually. Here we were chasing down kaun sa amino acid hai, kya hai, and our data support the idea that if you have a big change in the bias such that you're now sampling mutations which you have not sampled before in wild type E. coli, you will get a benefit simply because you have not sampled it. It's not because transversions are generally more beneficial, okay? What we're suggesting is that if you have an organism which is transversion biased and you force it to sample more transitions, you'll see the same effect. Okay? Got it? Kavita?
Yeah, so I was thinking, I mean, because of genetic drift, right? Suppose I have a very large population, I get stuck and they're called deterministic. And this gets stuck very fast. I'm again thinking of rugged landscapes. This is the work that I had in the archive. So, but if you have a smaller population, it's more stochastic, it samples more, and basically your fitness increases for longer time. So, what about genetic drift? Does that also play a role here?
We haven't explored cases of drift. This is largely in. Yeah.
Actually, you have small populations, I guess. I need to see the.
I'll send you the, So she's also done whole population simulations later to ask these questions as well, and same thing plays out there as well. So when you allow multiple beneficial mutations to also coexist, the effects are weaker, but they're still the same effects. So I'll send you the stuff, we can look at the details. Okay, so now can we have some other support for this, right? So far what I've shown you is just one strain of a transversion bias. What about other cases? Can we make transversion bias in different ways? And so these are very new data that we have put together recently. So here's a situation where this deletion of NTH-NEI, 2 genes, leads to a transition bias, okay? And the pink is a transversion bias strain. This is what you already seen, but we added some more data now. And you see that the same thing happens here. The gray one is shifted to the left compared to the pink, which is shifted to the right. And here's an even more stark situation where we delete another gene, MUT-H. It allows a very strong transition bias. And the MUT-T strain, which has a very strong transversion bias, you see the same difference in the DFEs, right? Anytime you're sampling more transversions, you are allowing the DFE to be shifted to the right. You have access to more beneficial mutations, okay? So now what happens when you allow the joint effect of mutation bias and rate to dictate what happens with mutators? Everything I told you so far, we were ignoring the fact that the mutator can have a faster rate of evolution, right? Because we were stopping at the one single mutation step point. Now we allow those higher mutation rates to also play a role. What happens then? And so Lindy has done further simulations to ask this question. And what you see is that, so in these simulations now, these are whole population simulations where there's no adaptive walking necessarily. Different mutations can come up and the better ones have a higher chance of fixing and so on. In these simulations, she introduces a mutator, okay, with a particular bias shift, a particular rate shift, and so on, and asks how successful is it in invading the wild type population? Okay, that's the question. And so these kinds of invasion analyses are also useful ways of asking what is going to be the evolutionary fate of a certain allele. If it's able to invade, it means it's successful, right? So here's the invasion probability on the y-axis. And there's 2 sets of simulations here. One's with epistasis, which mean that different sites can interact to determine fitness. So mutations at 2 different sites can influence fitness together. And red is the situation with no epistasis, with epistasis, and blue is without epistasis. Here's the starting bias, 0.9, so very extreme bias at the beginning. And then the new bias can shift in different places, okay? What do you find? You find that overall, the probability of invasion reduces as the new bias shift is very close to the existing bias shift, okay? Or new bias shift is close to existing bias. And there's very low probability of invasion if your bias is going in the opposite direction to what existed already here, and making it more extreme. But the closer you move to the opposite bias, which means reversing the existing bias, the higher your invasion probability. Okay, and this kind of pattern they observe at different mutator with different mutators simulating different increases in mutation rate, 10-fold, 100-fold, whatever. Okay. So what about experiments? And so this is again very new data from the lab. We decided to test this idea with different competition experiments in the lab. And so here's what we start with. The mutation rate, the wild type is here. We have a 10-fold increase, we have a 100-fold increase in mutation rate with 4 different sets of mutators. And you've seen some data on all of these in the last few slides. This left side is case where there's more transitions, right side is more transversions. And so now we can tease apart the effect of mutation rate and bias, because these guys are paired at the same mutation rate. These guys are paired at the same mutation rate, but they have opposite bias, right? So what is the prediction here? The prediction is that these guys should be the fastest at invading because their mutation rate is super high, and they should really kick out wild type pretty quickly, right? Because they have higher access to beneficial mutations, bigger access, faster access. These guys should be a bit slower than these because they have a slightly lower mutation rate, right? But we expect all these mutators to eventually kick out the wild type and invade the wild type simply because of mutation rate benefit. However, within a given mutation rate category, we expect the ones on the right to do better because they are sampling transversions more, which we predict are more beneficial. Everybody with me? How do we do the experiments? We have a wild type, a marked strain of wild type, which has a neutral phenotypic marker based on which we can measure things, its frequency in the population. We give it a starting advantage with 95%, and only 5% of each of these mutators in turn gets added in. And then we mix it up and we keep transferring the cultures every day, okay, in glucose media. And then we measure the wild type and mutator populations proportions at different time points during this trajectory, where we are asking how much mutator is increasing, how fast is it increasing. Like I said, we expect this kind of trajectory for all of them because they have a higher mutation rate, and we expect to see a benefit in this short-term experiment of about a month. And how do we measure mutator proportions? We measure it 2 ways, either using qPCR by measuring the frequency of the marker which we have in the wild type, or we can also use that phenotypic marker to plate on agar plates and ask what proportion of mutator colonies do we see. All right, everybody with me? Good, so what do we actually see? So here's a case where we have, and so we have 6 different replicates of each competition. You see everybody starts from roughly 5%, which is where we expect them to start. And in the low mutation rate situation, with NTH-NEI, which is transition biased, you find that only one population has fixed by 60 days, okay? And all the other populations have either gone, the population hasn't fixed, sorry, the mutator has fixed in one population. In 1 or 2 cases, the mutator has gone extinct, okay? And a few other cases where it's sort of hovering in between, not able to really fix yet. But in the MUT-Y case, poof, it goes up right away in every replicate, and it's fixed very, very quickly, right? What happens with the high mutation rate strains? They fix really fast, they fix very parallelly, and there's a small difference between H and T, where the case where we have more transversions fixes a little bit faster. Okay, and here's a way to see that a bit more easily. We can also ask, in those early trajectories, do we see a difference? So time to frequency of 20% of the mutator. It's an arbitrary fraction more or less, but it's useful for us to understand a few things. And you see that the MUT-T strain is the fastest to get to 20%, so it takes fewer days to get there. The H population here takes a bit longer. Ignore this for now. And then the other strain, which is also transversion biased but has a lower mutation rate, takes longer as expected, right? And then this is the time to get to a frequency of 95%, where we're saying they're close to fixation. And that again is much faster for the MUT-T compared to MUT-H. And of course, Y and NTH-NEI are much slower. And Y is, again, many lines do fix in the Y. Many lines don't even fix here at all. right? So all of this is supporting what we predicted, that mutation rate as well as bias both play a role in the success of mutators in these experiments. Any questions? Okay, so why do I think this is exciting or interesting? What we're suggesting is the evolutionary history of a population is important to understand the evolution of mutation bias. It's not that mutation bias is driven by selection for specific kinds of mutations or not. We don't see evidence of it. We don't need to invoke that at all. It's a simple statistical sampling question. Which one have you sampled before and which one can you sample now? If you like mangoes and you want to find new varieties of mangoes, you better go to new parts of the country where you'll find new mangoes. If you're going to be stuck in one city, you're not going to find as many new kinds of mangoes. And this is interesting because it allows people to now understand a lot better what is the role of the generation of mutations itself and the type of mutations itself in driving adaptation in different ways. Okay, so mutation rate and spectrum can jointly determine adaptive dynamics. I'll end by thanking everybody who's been critical for this work. So Mridula is a fantastic PhD student and now postdoc in the lab who has been driving this project. So it's all originated from her thesis. Here she is. Bhumika was an undergrad from IIC who came and helped Mridula collect some of the data I showed you. I didn't actually talk about Gaurav's work. He's a former PhD student who did some phylogenetic analyses to ask whether in the phylogeny of bacteria, can we see evidence of rapid gain and loss of DNA repair enzymes such that they would lead to these bias shifts back and forth. And Lindy is the mathematician who's been collaborating with us to understand all of this. Pratyush was a master's student, thesis project student in the lab who did the competition experiments initially. And later Shazia and Mridula have been doing the competition experiments as well as the DFE data that I showed you later. So it's a big team of people. I'll also say that we do many different kinds of projects. You've seen some of them, I think, in the posters yesterday. We work with beetles, we work with microbiomes, we work with bacteria to understand molecular evolution. If you're interested in joining the lab, please come find me or encourage other people to find me. Thank you. Right. Sorry? Mine? No, I'm done.
Okay, so we should thank
All right, any other questions? We have maybe a couple minutes.
The organism has a bias towards transition, and because of that bias, it has exhausted the sample space of the patient.
Not quite exhausted, but yeah.
Compared to this,
Yes.
which can be explored by transversions, the space that can be explored by transitions has been relatively exhausted.
Yes. Yes.
So introducing the reverse bias would help it explore the unexplored space and
Exactly.
mainly
Yes.
mutations, right? Yes. So if I were to use the mutant, which explores the transversion space and let it grow for a long time, and then do the reverse switch, would you expect to see the same
Yes.
jump?
Yes, absolutely.
So if it has been a random process, then why one of the first plots that was shown where the original taxa are always left to the bias?
Yeah.
Why is that?
I don't have an answer for that. I wish I did. So my current vague feeling is that there is some biochemical or some such barrier that prevents more transversions from either fixing or being sampled in the population as a starting point, yeah. And it's not clear to me why that is the case, right? So it may be a simple biophysical kind of constraint, right? Nothing else is going on. Transversions are simply not sampled for that reason. And I was very happy to find some old literature from the structural biology crowd where they seem to suggest that the DNA structure gets more changed in ways due to transversions, which are not stable, and so they're less likely to occur, etc. But turns out that's all like mostly air. Like later papers don't sort of find support for this, and there doesn't seem to be a consensus that that's happening. And so I need to catch hold of some more people who've done this kind of work to ask, like, tell me, is this right or wrong? And is that effect strong enough to explain this very strong transition bias that we see? But I don't have that kind of information yet, so I don't know what it is. What I think our work does help to understand is that variation on the left side. Okay, given everybody's on the left side, why are some more left and why are some more right? And we predict that that shifting is going to end up evolving under selection over time. So lineages should be on some side and then sort of drift a little bit on this side maybe, but selection should draw them up again, pull them up again on the other side. Yeah. But yes, good. I'm glad that it got through. At least one person has gotten it. Any other questions? All right, thank you very much.